And good morning. How's everybody doing? Dr. Chris Roddenberry coming to you from the University of Rijeka Library here in Rijeka, Croatia. This week is Chapter 5, Sensation and Perception. How are you doing, Angelina? Good to see you. Thanks for coming today. I hope we have some other people in the live stream broadcast. Looks like we don't. Maybe it's just you and me today, Lena, so we'll just have fun talking by ourselves, okay? One thing I want to show you before we get started, did you see on the announcements page today, did you see what I sent you? I'll show you, it's really cool. Where's right there? Pretty cool, isn't it? That is what's known as an Ames room. That's my friend Dale and I. We are actually the same size. There is no photographic trickery. That's just a room that is out of square, that when two people stand in corners, it gives them the illusion of being very different sizes. Pretty hard to believe that your brain can get fooled by that, don't you think, Lena? To me, that's like the craziest thing I could ever imagine. And you know, the thing is, no matter how hard you look at it, no matter how much time you spend focusing on it, it's still, your brain just can't reinterpret that stuff correctly. And so we're going to be talking about the sense of vision today. And one of the things that I'm going to talk a little bit about is visual illusions. And tomorrow night in class, when we do the webinar, I'll show you some more visual illusions and we'll have a little bit of fun thinking about these kinds of things, okay? So let's go ahead and get started with today's lecture, Sensation and Perception. Today we're going to talk about vision. Scientists actually have studied vision the longest, know the most about vision. And so we're going to spend an entire day talking about the visual process. And then tomorrow we're going to talk about the other senses that we have, okay? So let's go ahead and get started. If anybody else is in the broadcast, go ahead and type in a hello and let me know you're here. Otherwise, it'll just be me and Alina today. Now, sensation and perception actually is the first area of modern psychology. Do you remember in week 1 when I talked about Gustav Fechner, the first to demonstrate that the mind could be measured? I said he was the founder of scientific psychology. He was actually interested in sensation and perception. And so the branch of psychology that's interested in sensation and perception is actually called experimental psychology. Not because they're the only ones that do experiments, but they were the first people to do experiments. So experimental psychology was what Gustav Fichtner did. And he actually noticed this very interesting phenomenon he called the just noticeable difference. In fact, it turns out that there's a very systematic relationship between the amount of change in a sensory stimulus and your ability to detect that change. So the amount that something has to change for you to notice it is called the just noticeable difference. And whether you're talking about sound, sight, weight, or any other sensory stimulus, there is a very specific amount that thing has to change for you to be able to detect an amount of change. And this little graph right underneath me is a demonstration of this thing called a just noticeable difference. Now, his area would eventually become the modern subfield of what we call experimental psychology, the study of consciousness in the human senses. And we're going to be talking about sort of 5 and more senses this week. We're going to be talking, of course, about the ones you can think of: vision, hearing, smell, taste, touch. But you know what? There are other senses too. There's a sense of balance. There's a sense of pain. There's a sense of warmth. And your body has sensory organs all over that take information about what's happening in your body and convey it to your brain in terms of some sort of sensory perception. The purpose of our senses is to provide information about the environment that our brain can then use to evaluate and change our behavior in order to achieve good senses and avoid bad sensory experiences, right? Okay, now the lecture, the point that I'm going to try to make to you this week is the difference between what we call sensation and perception. Okay, sensation is what happens when light goes into your eyes and causes those your rods and cones to activate. Sensation is what happens when sound goes in your ears and causes your little hair cells to vibrate. Sensation is what happens when chemicals go in your nose and cause your nerve cells there to activate. Sensation is the activation of the sensory neurons out around your body. Perception, on the other hand, is integration of these sensory experiences and you being able to perceive what it is you're looking at. So a visual illusion, kind of like the Ames room that I just showed you, okay? So you're seeing the color of our shirts, you're seeing my face, you're seeing Dale's face, you're seeing two of us, and that is the sensory experience. But then your brain perceives us to be much different. That's the second process. And visual illusions work because the sensory information that we get into our brain tricks us into imagining that we see something else. Oh, that we see something else. Okay? And so what I want you to be smart enough to know is the difference between sensation, the activation of your neurons, and perception, you figuring it out, what it is you see, what it is you hear, what it is you smell, or what it is you taste. And so I just want you to know that it's a two-step process. All right, now we have these cells. I talked to you about sensory neurons that bring information to your central nervous system, and then I talked to you about motor neurons that take information from your central nervous system to the rest of your body. But there's a third kind of neuron that we're going to talk about this week. It's called the sensory receptor cell. Now, the sensory receptor cell is at the periphery of your body, and the sensory receptor cell, its job is to turn physical stimulation into a series of action potentials. So when light goes in your eyes, it hits these pigments called opsins, and these opsins cause your eyeball, the neurons in your eyeball, to generate action potentials. That process is called transduction, and it occurs in the receptor cells, okay? So you have neurons, sensory receptor cells on your hand. And when you touch your hand and put that mechanical pressure on your hand, that mechanical pressure causes these neurons to generate action potentials. The neurons that generate these action potentials based on this mechanical energy are called receptor cells. And the process of turning physical energy into patterns of action potentials is called transduction, okay? Now, there are lots of different sensory receptor sites. There are mechanoreceptors. So you have hair cells in your ear that bend when sound waves cause them to push over. You have touch receptors and pain receptors and stretch receptors, and all of these respond to mechanical information, physical stimulation that pushes these neurons. Those are called mechanoreceptors. In your tongue and in your nose, you have chemoreceptors, and those receptors are responsible for turning chemicals into action potentials, okay? And then in your eyes, you have photoreceptors or visual receptors, and their job is to turn photons of light into action potentials. And then you also have thermoreceptors that judge how hot different parts of your body are. And you have nociceptors, which are receptor cells that turn mechanical injury into the feeling of pain. Now, a couple of weeks ago, we talked about chapter 2, when we were talking about parts of the brain, I introduced you to a part of the brain called the thalamus. It's right down in the middle of your brain, and it's responsible for helping you have a conscious sense of all of these senses. So when you smell something, when you see something, when you see something, when you hear something, not when you smell something, all of this information is routed through this brain structure called the thalamus. All of your conscious sensory experiences, with the exception of smell, because that seems to have developed before the thalamus developed, all of these sensory experiences go through your thalamus. And I'm going to want you to know that for this next exam, okay? The thalamus. It's sort of a sensory way station where all of your senses travel as they are going to the sensory cortices. And if you remember, looking at this picture down here, when we were talking about areas of the brain, I introduced you to the different sensory cortices in the brain. And I'm going to want you to know those for this exam. Okay, so your olfactory and your gustatory cortex, your taste and your smell, are processed in the frontal lobe of the brain. Okay, your auditory cortex, where hearing is processed, is in your temporal lobe. Okay, and then your sense of touch is processed in the primary somatosensory cortex, okay, located in your parietal lobe. And then your vision is processed in the primary visual cortex, which is way in the back of your brain in the occipital lobe. And your cerebellum, which is not on this picture, which is right below your cerebral cortex, is where your sense of balance is processed. So on the exam, I may ask you to tell me where these different sensory cortices are located. Okay? Let's see. Do we have anybody else here? No, just you and me, Lena. Okay. Well, that's fine too. Let's get back to it. All right. So let's talk about okay, and then again, I mentioned earlier sensation and perception. Keep in mind that when information is brought into your brain, that's sensation. But then your brain has to put all that information together and decide what it is you are looking at. And that process is called perception. Okay? And perception can be fooled. I can cause you to feel things that aren't there. I can cause you to see things that aren't there. We can even cause you to smell things that aren't there. Okay? And so I do want you to know the difference between sensation and perception. Okay? Let's talk about vision, the anatomy of an eyeball. So your eyeball is responsible for processing light energy and turning it into the sense of vision. Okay? Now let's look quickly at this. I don't want you to know a ton about this eyeball, except I do want you to know that you have an iris, which is that pretty colored band of tissue. I have a blue iris, some of you have brown irises, some of you have green irises, and that's just a band of muscle that can expand and contract to regulate the amount of light that comes in your eye. I don't know if you've ever been to a movie, Lena, and you come out of the movie theater. You know how it's really dark in the movie theater? And as soon as you come out, the world seems super duper bright and it's very uncomfortable for your eyes. Well, that's because when you were in the dark movie theater, your iris opened way up and expanded your pupil. You had that big black dot in the middle of your eye so that it could get more light into the eyeball. And then when you come out of the movie theater, your iris is too big and you getting too much light in your eye and it's not comfortable. And it takes about 30 seconds for your iris to shrink a little bit and regulate the amount of light that comes into your eye. Now that black dot in the middle of your iris is not a black dot, it's actually a hole. And if you took a flashlight and looked in somebody's pupil, which is the black dot in the middle of the eye, you would actually be able to see the back of their eyeball. When you go to an optometrist and they shine that light in your eye, they're actually looking at the back of your eyeball called your retina. okay? And your retina is where your photoreceptors are. Two other things I did want to mention. You actually have right behind that band of muscles, there's actually a transparent piece of tissue called the lens. And just like a pair of glasses, okay, this lens is responsible for focusing the light energy perfectly on the back of your eyeball, on your retina. Now, some people's eyeballs aren't shaped perfectly or their lens isn't shaped right, and they have to go to the doctor and get corrective lenses. Lena, do you wear contact lenses or glasses? These actually are corrective sunglasses because my eyes don't see very well. And basically what prescription eyeglasses do is they help your lens focus the information right on the back of your eyeball. Okay, you're like me, Lena, you're a glass wearer too. And one other thing I do want to tell you, you're not old enough for this, Angelina, but me and my wife are. You actually have these muscles on the sides of your lens called ciliary muscles, and what they do is they can stretch and cause that lens to contract and change shape. And so what happens is when you're looking at something close, if you move real close up to the screen and look at what's on the screen, you're going to notice you feel a little pressure in your eyes. That's your ciliary muscle adjusting the shape of your lens so that you can see perfectly when you are doing close-up vision. But when you get my age, that lens muscle, just like the rest of the muscles in your body, that lens gets a little bit inflexible, and the ciliary muscle can't bend it. And that's why older folks have to wear bifocals. And they only need the bifocals when they're reading because their ciliary muscle in their lens can't flex to help them put images directly on their retina, and things get blurry looking to them. So that's all I really want you to know. I'm sorry this complicated picture with all kinds of different parts on it. Don't worry about them. I just wanted you to see the pupil, the lens, and the ciliary muscles. What we're really going to focus on is the back of the eyeball, which is called the retina. And the retina is neural tissue that contains the visual receptor cells called rods and cones. You have two different types of visual cells in the back of your eyeball. And the retina is where those nerve cells are. And there's a specific part of your retina called the fovea, okay? And when you are looking directly at me, my image is falling on your fovea, right? And the fovea actually has less blood cells because it's so packed with visual receptor cells. And so when you're looking at something, that's where you're going to see it the clearest. These things are out here in the periphery of your vision, look kind of blurry, and that's because the images of these objects aren't falling directly on this fovea part of your eyeball. And it turns out you don't realize this because most people don't look at their peripheral field of vision, but you really only have clear, crisp vision in the middle part of your visual field. Right? Now, there's also one other structure I want to talk about. It's called the optic disc or the blind spot. And this is the part of your retina where you do not have any visual receptor cells because this is the hole out the back of your eyeball where all these nerve fibers begin go to the back of your brain. It's where your optic nerve begins. Now, why don't we have a hole, a blind spot in our field of vision? Excuse me. Why? Because you have two eyes, and each eye is covering the other blind spot. Thank you, Angelina. Each eye is covering the other blind spot. Now, you might say, well, Chris, even when I close my eyes, I don't see a hole in the field of my vision. And you know what? That's right. That's because your brain actually guesses what's in the spot in the middle of your vision. There's that little bitty blank hole in your vision where you're not getting any feedback, but your brain fills in the missing part of your visual field. Okay, so let's move on from the anatomy of an eyeball. Okay, now I want to talk specifically about the retina and the cells in the retina. You have these two kinds of visual receptor cells, and these visual receptor cells are sometimes called photoreceptors, and that's because they are sensitive to stimulation by light photons, little packets of light called photons. All right. Now, if you look right underneath me, you've got these two cells, rods and cones. I want you to notice one important thing about them. Your rods and cones are actually facing away from what you're looking at. So if I'm looking right at you, that means my rods and cones are actually facing the other way. What they do is the light photons go through your eyeball, go through those rods and cones, and it hits the back of your eyeball. And then your rods and cones see this light energy as it bounces off the back of your eyeball. So your rods and cones are actually facing backwards. All right. Now, let's talk about the differences between these things. So you have rod cells and you have cone cells. Why do you have two different kinds of cells? Well, these two different kinds of cone cells allow you to both see well at night and also allow you to see things in three colors or see all the beautiful color combinations that you see. So your rod cells, if you look over here, I've got the picture, see the rod cells kind of look rodish. They look like a staff or a rod or something. And then your cone cells kind of have this pointy end, right? Now your rod cells contain an opsin, rhodopsin, that is very, very, very sensitive to light. It doesn't take very many photons to activate this chemical called rhodopsin. And when rhodopsin is stimulated, it causes an action potential so that you can see something. Now, your rod cells are located mostly outside of your fovea. They're in your peripheral vision. They don't need a lot of light. They're much more sensitive than cone cells, and they are responsible for letting us see at night, but they don't see color. Okay? Now, on the other hand, cone cells are located almost exclusively in the fovea, the middle of your eye. Now, these cells contain three different kinds of opsins, but these opsins are not very sensitive. They need more light to activate action potentials, okay? So your cone cells don't see as well at night, okay? And however, there are three different kinds of opsins that create your color vision. So you have one opsin that sees sees red colors. You have one opsin that sees a blue color, and one opsin that sees green colors. Okay, so you have three different opsins. Now, what's going to probably blow your mind is that you only have color vision in the very middle of your field of vision. Now you might say, Dr. Roddenberry, I see color in the periphery. Your brain adds the color in the periphery in after the information gets to your brain. So although I'm looking at this wall, and this wall in front of me is yellow, and all I can see is the middle part of my eye sees yellow right in the middle of my field of vision, and then my brain knows that the rest of that wall is yellow. So my brain actually adds in that color. So color vision in your periphery is actually a perceptual process, not a sensory process. Okay, now, if you'll notice in this picture, you've got rods and cones, and then you've got these things called ganglion cells. And ganglion cells turn cones and rods off so that so that they work better. So when you see a black line, an edge, the part of your brain that sees the black line is activated, and then it deactivates the black seeing cone right beside it so that lines look sharp. You ever notice that you can't see red-green. There is no such thing as reddish-green. And the deal is, when your red cone cells are activated, the ganglion cell actually turns off the green cone so that it can't send information, so that you only see red and not green. All right? Now, the weird thing is, there's this thing called an after image. So I don't have it right here, but if you were to look at a red dot for about 30 seconds and just stare at that red dot very hard, right? And then look off to a white wall, what you would see is you would see a light image of a green circle. And that's because when the red goes away, your ganglion cell lets off of the green cone, and the green cone actually bounces back a little bit and creates the after image of green. Okay? So I want you to know rod cells, very sensitive to light, mostly in the peripheral part of your retina, and they give you night vision, but they don't see in color. On the other hand, you have cone cells that are located almost exclusively in the middle of your retina, where things are when you're looking at them, and they are less sensitive to light. They need more energy to work. However, they give you the sense of color vision because you have 3 different types of cone cells. And then the ganglion cells work as sort of support structures, and they condense the information from your rods and cones before it leaves your brain. Turns out you have 10 million rods and cones, but you only have 1 million fibers leaving your eye. So the information that's collected by your eye, before it even leaves your eyes, it's condensed by 10 times down to less information before it even leaves your eyeball. Okay? Now, quickly, for some reason, my colors, when I do this, it doesn't go right, but that's supposed to be green in the middle. It looks black to you, but I swear it's green. And here's how color vision works, okay? So we have these opsins, light-sensitive proteins in their eye that cause action potentials to be generated. Now, what you're looking at in this picture over here is what's called the visual light spectrum. So your eyes are sensitive to light energy and of 400 from a wavelength of 400 to a wavelength of 700. This is called the visual light spectrum. So 400 nanometers to 700 nanometers. Now, X-rays are light energy that are above or are outside your visual spectrum. Infrared light is light energy that is outside of the visual spectrum. So you can't see X-rays, you can't see heat signatures of animals. You can instead see this light coming in this visual light spectrum. Now your rods and cones your rods activate equally no matter what wavelength light they're seeing in this visual light spectrum. However, your cone cells are differentially sensitive to different parts of the wavelength. So your short cones are most sensitive to wavelengths in the shorter end of the visible spectrum. And this gives you the perception of blue, all right? On the other hand, you have your red cones, and they are more sensitive to light at the other end of the visual spectrum, close to 700 nanometers. And these cones give you the perception of red. Okay, and then you have some cone cells that are most sensitive to wavelengths in the middle of the visual spectrum, and that gives you the perception of green. Now, I don't know if Angelina, if you're colorblind or if you know anybody who's colorblind, but people are colorblind because they have a mutation in their DNA that causes them not to create the proteins that allow them to differentiate, usually between red and green. So they're missing a protein in their eye, so they don't see this difference between red and green. Quick question, we got a couple of people in the audience. If I told you that dogs were completely colorblind and they saw really well at night. Dogs have great night vision, but they don't see the world in color. They see the world in black and white. What kinds of visual receptor cells do you think dogs don't have? I'd be curious to see if you can figure that out. What kinds of visual receptors do dogs not have? And I'll go on while I wait for the answer. They don't have cone cells. They don't have the cone cells, but they're going to have plenty of rod cells. Now, what I'd like to do is talk a little bit about how information travels from your eyeball all the way to the back of your eye. All right? Now, here's the deal. You have an optic nerve that leaves your eyes. And basically, the optic nerve is a collection of neurons that go from your eyeball towards the back of your brain. So they're going to run the whole length of your brain from your eyeballs all the way to the back of your brain. So a couple of things I want you to notice here. If you look at this picture underneath me, what you see is a brain, and you see these little red and blue lines going from the eyeballs all the way to the back of the brain. Now, notice that this person is looking out and they have a field of, you notice that out in front of this weird eyeball looking thing, you see a red patch and a blue patch, and they kind of overlap a little bit. Now, what this means is that each of your eyeballs is looking at half of your visual field, both halves of your visual field. So the right half of my right eyeball sees the left half of my visual world. The right half of my left eyeball also sees my left visual world. And the left half of my left eyeball sees my right visual world, and the left half of my right eye sees my right visual world. So in a sense, each eyeball sees the left half of the world and the right half of the world. And you'll notice that these two visual areas overlap, and that's probably right where the fovea is, all right? Now, here's the thing. What we do know is that the left half of your brain sees the right half of your visual world, your right hemifield, and the right half of your brain sees the left half of your visual field. So what we have to do is we have to switch the information coming from the eyes so that the right half of the visual field in the right eye and the right half of the visual field in the left eye go to the left part of your brain. And the left half of your visual field from your left eye and the left half of the visual field from your right eye goes to your right part of your brain. This is accomplished at a structure called the optic chiasm. Okay, this is where information from each field comes together to be transmitted to the correct hemisphere of the brain for processing, the optic chiasm. Now, your optic tract nerves are then going to synapse in your thalamus with a different set of neuron that carries it all the way to your visual cortex. And if you look at this picture below, you'll notice at the very back, the left primary visual cortex is in blue, and the right visual primary cortex is in red. And you'll see that the right red visual cortex corresponds to the left field of vision, and the blue left primary cortex corresponds to the blue color on the right hemifield, the right field of vision. Okay? Now, the nerve comes all the way back to the thalamus, synapse is there, and then a different nerve takes it all the way back to your visual cortex. Remember, all of your senses, except for smell, travel through this thalamus structure. Now, I do want you to know there's another structure in your in your thalamus called the superior colliculus. And this is the structure that connects your vision to your other senses. So when I hear a sound, my eyes know where to turn to see that sound that was just made. In order to grab this water bottle, I have to be able to guide my hands to that water bottle. So the superior colliculus is the area of your thalamus that ties your vision to your other senses. Okay? Your superior colliculus. And just as a side note, I don't know if you remember this, but when we were in chapter 3 talking about sleeping and waking, I told you that some of these nerves coming from your eyeball, they don't go all the way to the back of your brain. Instead, they go to this suprachiasmatic nucleus that is in the hypothalamus. Hypo means below, so the hypothalamus is a structure right underneath the thalamus. So I think it's about 20% of your nerve fibers actually go to your hypothalamus and help you know when it's night or day, all right? But 80% of them are going to go to the thalamus and then travel all the way back to your primary visual cortex, located at the back of the brain. Now, it's called the primary visual cortex because it's where the primary aspects of a visual stimulus are processed. What are the lines? What is where is that object located? Where is that object moving? What colors are on that object? So the primary aspects of visual stimuli are processed here. But then your brain's going to put those together and start making shapes and objects in what we call the visual association cortex. And I'm not sure if I have a picture. Let me see if I can go back to where is it? Right here. Okay, so you'll notice that the primary visual cortex is right there at the back. That's that orange area. But do you see that yellow area outside of the primary visual cortex? That's called the visual association cortex. And that's where these different parts of vision begin being put together so that you can see shapes and objects. And then that information is going to travel to your temporal lobe where you remember what those objects and shapes are. There are people who suffer from what we call visual agnosia. They can't, when they look at an object, they can see the lines and the colors, but they can't tell what object they're actually looking at. And so, again, as I suggested to you, the fact that you see colors might be a sensation, but the fact that you can put those things together and know that you're looking at an apple is perception. Perception. Now, let's talk a little bit about perception for the next couple of minutes. Vision is sort of a learned ability. Your brain process has those has those rods and cones in your retina, and they process light and dark and color and lines and movement. But then your brain learns over time how to interpret patterns of stimuli in order to make sense of your world. And that's the beginning of this process we call perception. Now, way back, almost 100 years ago, these German psychologists identified sort of 5 basic patterns that our brain uses to group and organize things in order for us to begin understanding our world. So, for example, when we see things, we tend to group them into things that are close together. So if you look all the way over in A, these groups are grouped together as an objects. So instead of seeing 16 dots, you're more likely to see 3 groups of dots, okay? If you look in B, what our brain does, our brain is deciding that we're looking at 2 objects, and a bunch of straight lines and a bunch of squiggly lines. So your brain can differentiate between those 2 sets of objects. And so we say that they are separate from one another. So we have the red lines and the black squiggly lines. Now, it's see-through, so you can see what's happening. It's going there. But when you were to look at this object, if that pink can was completely solid, you wouldn't say that's a pink can with 2 black squares on it. Your brain would guess that the object, the black object, is just one thing that goes behind that can. And so that's what we would, that's sort of the perceptual quality of perception we called continuity. Now, if you look at the 2 shapes down here, you notice that in D, you actually have a triangle. To be truthful, that's not actually a triangle. It's 3 arrow-looking things that just happen to be oriented away from one another. But your brain decides that that looks like a triangle. And then the same thing happens with contours. Instead of seeing 4 Cookie Monster Pac-Mans, your brain actually sees the square in the middle of those objects. That's called the perceptual clue of illusory contours. Now, Lena, if you've ever been outside on a nice cloudy day and a nice beautiful day and looked up at the clouds and you've decided, hey, that cloud looks like a dog, that cloud looks like an elephant. Your brain, in a sense, is trying to figure out, based on similar shapes, it's trying to figure out what in the heck it is looking at. And our brains really like to impose order and meaning on objects. And that's what your brain's doing when you're out looking at clouds on a sunny day and think you see shapes and animals and whatnot. Now, we also have perceptual cues that allow us to see vision. Now, here's the thing. We have 2 kinds of depth perception. The first is created because we have 2 eyes, and these 2 eyes are seeing the same object from slightly different angles. So the left eye sees Chris, sees my computer, and my right eye sees my computer, and my left eye sees my computer. But they have slightly different images because they're looking from different locations. This is what's called binocular disparity. And your brain reads the muscle power that you exert. So the closer things come to you, the more your eyes turn inward, and your brain reads that turning of the muscles, and over time learns to judge depth based upon how much the eyes are turning in. That's called that, and the fact that the 2 images are a little bit different. So it's the muscle cue, the stretch of the muscle, as well as the different images. Over time, your brain learns to interpret them and figure out how far objects are away from you. Now, not only do we do that, but your brain learns that the world has sort of these patterns that you can figure out after a while. So, am I in front of the phone, or is the phone in front of me? Well, you're going to say, of course, the phone is in front of me. Why do you say that? Because you can't see all of me, but you can see all of the phone, right? That's a monocular depth cue, a monocular depth cue, okay? We call that occlusion. A nearer object blocks an object behind it, all right? And so we can still tell depth even if we only have 1 eye, right? And if you've ever seen anybody who's painted a painting, they can use these depth cues to create the illusion of depth. There's the other one called height in the visual field. Objects that are lower in the visual field are seen as nearer than objects that are higher in the visual field. Our brain interprets that as distance. Relative size. Here's the deal. Objects that are farther away, the man on the sidewalk, project a smaller retinal image than close objects of a similar size. So I know that men in general are about the same height, but the guy back at standing on the sidewalk at the back of the crosswalk looks teeny tiny, and this guy in the white shirt with the red tie, he looks a lot bigger. And since your brain knows that one object, that these 2 objects should be the same size, it tells you that the guy who has a bigger image on your retina should be bigger, okay? And then so we've got familiar size, then we've got linear perspective. Typically parallel lines, if you look at them as they go off into the distance, they don't run parallel, they will actually run towards one another. And so seemingly parallel lines that converge give your brain the clue that the lines are going away from you. And then you'll notice if you look at this sidewalk, there's more distance between the white lines close up to you and less distance between the white lines farther away from you. This change in the texture gradient is interpreted by your brain as a cue to depth. Now, here's the deal. Let's go back to one time and let's talk about this this visual illusion right here. How does this visual illusion work? Well, the thing with an Ames room is that this Ames room is all out of square, all right? And your brain has learned that walls of a house are always parallel. But this room is actually not square. It's more like a trapezoidal shape. And it's also out, it's not facing directly at you, but I'm actually farther away from you uh, than Dale is. And because my wall is taller, your brain interprets me, it assumes that I must be shorter, even though we're the same size, but I'm standing farther away and my wall is much taller. And your brain interprets these cues and realizes and thinks that I must be smaller. It makes the judgment that I must be smaller than Dale. Okay. uh, Lena, that's all I had to say today. I think we have one more person in the audience, though you haven't chatted, and that's fine. But that's really all I wanted to talk about with vision today. Don't forget, tomorrow we're doing the lecture. The Thursday lecture will actually be tomorrow. So our second lecture of the week is tomorrow morning, Lena, if you're coming. And don't forget, we do have our webinar tomorrow night at 7 p.m. And we're going to talk about visual illusions and the perception of vision tomorrow. Okay? Do you have any questions? Any questions? I'd like to thank you for coming. Have a great day. And Lena, I will see you as soon as I finish my next class, which should be probably 5 minutes to 11, maybe 11 o'clock. Okay? Take care and have a great day. Bye.